Hi, guys. So we're looking at meiosis and genetic diversity as our key focus in this unit. So we want to start off talking about heredity and genetics. So genetics is the study of heredity and hereditary variation. So genetics is all about the study. Heredity is the transmission of the traits from one generation to the next. So it's the blue eyes that your grandfather may have had that got passed on to your father that get passed on to you. That would be heredity. The studying, so when we talk about Punnett squares, when we talk about the gene sequences, when we're talking about how that happens, we're talking about genetics. And it's really hard sometimes to keep the two straight. So traits are passed from parent to offspring through genes. And so when we're talking about genes, remember, we're talking about that little segment of the chromosome of the DNA that codes for that specific thing, or combination of segments, because sometimes it's actually more than one section of different chromosomes that code for a specific factor. And so we know that offspring, of course, acquire the genes from their parents by inheriting the chromosomes. So you get half of your chromosomes from mom, you get half of your chromosomes from dad. And that's what makes you up. And so that's why sometimes you'll see more of one characteristic than another, just by whichever one's stronger within those passed on characteristics. And so when you're looking at a family group, you're often going to see certain characteristics that are the same for everyone, or at least similar. And so that's why you see similarities when it comes to families. Okay, the other big thing we want to talk about here is sexual versus asexual reproduction, because reproduction plays a big role in our genetic variability or diversity. How are the genes being passed on? So when we're talking about asexual, we're talking about a single individual or organism. And so remember, it's not just about human beings, although we tend to often focus our worlds on human beings, but we've got sponges, we've got spiders, we've got snakes, we've got plants, all these different things. There is no fusion of gametes. So we don't have an egg and a sperm, so they can't come together. In asexual reproduction, all of the organisms are basically clones. So this is a natural cloning process, because the offspring are, for the most part, an exact copy of parents. I say for the most part because we know, we learned about it when we talked about mitosis, how even when we go through this process, there could be slight errors or changes in DNA. And so those changes could ultimately lead to a slight variation, but for the most part, they are clones. Mutations, like I was just talking about, are going to be the only source of variation. Okay. And they can produce asexually through mitosis. So basically, when we're talking about asexual reproduction, we're talking about using the process of mitosis to make new individuals. With sexual reproduction, there are two parents, a male and a female. Remember, we've had this discussion that the reality is biology is designed as a binary system. The offspring are going to be unique combinations of genes from the two parents. Remember, you're getting half your genes from mom, half your genes from dad. And so based on that mixture, they're going to be unique to you. Genetically varied from parents and siblings. So just because your siblings also got half from mom and half from dad, they may not have gotten the exact same genes in that half and half. So there is still variation between parents, between siblings, etc. So when we're talking about this system, you've got to kind of remember this idea of homologous chromosomes. So the homologous chromosomes are the two that pair up. So we've got a paternal and a maternal. When we duplicate those for mitosis, remember that's when we came up with the homologous pair of chromosomes that had sister chromatids. The two matching, or the two copies. Okay, so homologous chromosomes, a pair of chromosomes, same size, same length, centromere position, that point where they connect, carry the same genetic information. So one homologous chromosome is inherited from mom and one is from dad. When we replicate those, we end up with a sister chromatid situation within our homologous pair. And then we have karyotypes. So with karyotypes, you can see how we take those homologous pairs that we have, and we're going to kind of match them up. So we have the one from mom, one from dad thing with number 1, 2, and we number them and we line them up based on sizes and shapes and genes and all of these things. This one, you'll notice, has 2 X's, so it happens to be a female. So on a karyotype, which is just simply a display of the chromosome pairs ordered by their size and length. So remember, we have our pair of homologous duplicated chromosomes. We've got 2 copies, one from mom, one from dad concept. So in an actual karyotype, sometimes it's really hard to see the sister chromatids in each pair. So you might not really clearly be able to tell that we have 2 exact copies. But a lot of times we work with, here we work with the illustrated ones. Okay. So if we're talking about, especially since we're focusing a lot in this unit and a lot of this study in humans, and of course in organisms that are going to reproduce sexually, you need to understand the difference in some of the cell terminology. So what we have are what we call somatic or body cells. So these are typically going to be diploid, or we call it 2N, which means we have 2 complete sets of each chromosome. We have one from mom, one from dad for every single chromosome. We haven't split them. We haven't reduced anything. So in humans, 2N is 46. That would be 23 pairs. And then there are gametic or sex cells. So these are typically going to be haploid, or we just call it N, and it's got 1 set of chromosomes. So think about this as what we're talking about here is the egg and the sperm. Humans, the sperm and eggs are going to be N23. So eukaryotes have DNA packaged in chromosomes, those little X-type structures. So there are 2 types of chromosomes. We have autosomes, that are chromosomes that are not determining sex. In humans, that's 22 pairs, so 1 through 22. And then there's the sex chromosomes, X and Y. Eggs are considered X. Sperm have X or Y because the males have both options. Males determine the sex of the individual. All sexually reproducing organisms have both diploid and haploid numbers because they're going to have somatic cells and they're going to have gametic cells. So within the life cycle, it's a sequence or stage of processes through reproduction. So notice in our little diagram, we have our baby, 2N, goes through mitosis and over and over and over as it grows and develops. So our adults are still 2N. But each of our individual adults are going to make gametes. So we have an egg for the female and a sperm for the male. Notice that these are haploid. They're N. When they join in the process of fertilization, when conception occurs, they create a zygote, which is 2N, which then will go through mitosis, developing until it becomes a baby that is born back to the cycle, back to the beginning of the cycle. So fertilization and meiosis alternate in sexual life cycles. So sometimes in some situations when they talk about life cycles, they're going to talk about alternation of generation. We don't normally do that, use that terminology with humans, but it comes up a lot, especially like with plants. So fertilization is when the sperm cell fuses with the egg and forms the zygote, just in case you missed and don't understand what fertilization is. Okay, so hopefully this will have helped you and you are ready for the next lesson. Bye.